Hello everyone, my name is Jean-Christophe Lachance. I am a fourth-year PhD student in the Department of Biology at the University of Sherbrooke, which is just about an hour east of Montreal. My presentation is titled Genome-scale modeling of Mesoplasma florum reveals key feature of a minimal gene set. The questions I will raise and try to answer during this talk are, what do we learn as we model the metabolism of such a small organism like Mesoplasma florum? And secondly, as we've modeled that organism, could we use this model to generate a prediction of a minimal gene set? If so, how accurate would that model-driven prediction actually be? So first, let's go back in time and try to explain from a historical perspective, why are we interested in studying minimal cells? So since the works of Mendel and Darwin, the question in biology has always been like, how does life work? So with the theory of evolution, we then started asking, what is the material support of the gene? So we now know this to be DNA since the works of Watson and Crick. And this discovery led to the elucidation of many molecular mechanisms happening within the cell, in an era that we here call the era of molecular biology. For example, it became possible to understand how the information contained in the DNA was expressed via the operand structure and was translated into functional units such as proteins via the genetic code. Understanding those molecular mechanisms allowed the development of tools to manipulate DNA, like restriction enzyme and polymerase chain reactions. The development of these tools were a paradigm shift in biology, as they allowed to start conceiving our own DNA assemblies to suit the purpose we want. Finally, with the development of sequencing technologies and the publication of whole genome sequences, came the era of genomic that overlaps with that of synthetic biology. While on the lower branch, many high-throughput sequencing projects provide an increasingly higher number of sequences to analyze, the upper branch managed to synthesize entire genomes from scratch and start making living organisms with synthetic genomes. Since new entities are created, we can say that the goal of this upper branch resembles that of an engineering discipline. While we get more and more sequences, we also get an increasing number of proteins with uncharacterized functions. This can be slightly problematic since many projects are geared towards obtaining more sequences, but without performing the biochemical characterization that is still a limiting factor for proper engineering in biology. This is to say that we have a great potential to address big challenges with the advent of synthetic biology, but if we can't establish proper design principles, we will always run into unexpected issues. As we just said, standards are essential for any engineering discipline since they ensure the reliability of the products that will be produced. To understand how minimal cells apply in that sense, we can draw the parallel between them and operating systems in informatics. An operating system in a computer is a standardized software that enables to implement applications. Its standard ensures the compatibility of the applications with its own structure. A minimal cell proposes to do something similar. It would contain the minimum number of genes necessary to sustain self-replicating life. Given its smaller size, the number of undefined components it contains would be lower, hence facilitating its entire characterization. In turn, full characterization would allow the establishment of design principles and compatibility with the pathways that would be added to it. In practice, Unix-based operating systems like Linux and Mac are compliant with the POSIX standard. In biology, similar initiatives already exist with the systems biology markup language that ensures standardization in computational modeling of biology, and the JCB ISN3 organism that is the first synthetic minimal organism. Given the similarities between both fields, we can expect that more minimal genomes will be designed and created in the future, and that design principles will emerge from these efforts. Mesoplasma florum is an organism from the Mollicutes phylogenetic group and is closely related to Mycoplasma mycoides, the natural parent of JCB ISN3. As for many Mollicutes, its genome size is much smaller than eukaryote or prokaryote, such as E. coli. Its natural genome is nevertheless bigger than that of JCB ISN3 by about 261,000 base pair and 207 genes. Given the phylogenetic proximity of the two Mollicutes, it becomes really interesting to consider what would happen if we were to reduce the genome of M. florum. How close would this reduced genome actually be from the synthetic cell? Some features that make M. florum interesting are also its fast doubling time of 40 minutes compared to more than 6 hours for many other Mollicutes, and the fact that it is a non-pathogenic organism that can be manipulated safely in a biosafety level 1 laboratory. Gladly, recent effort in our laboratory have allowed to gather a lot of experimental information on the mFloram cell that can be used for modeling. We actually are working to submit this paper rather soon, and we'll have it on bioRxiv, so I encourage you to go and have a look at it. Specifically, the information you will find in it are the physical properties of the cell, such as the cell weight, size, and density, the detailed biomass composition, including macromolecular wave fractions, composition of the polysaccharide layer, and finally, growth parameters such as doubling time. So before we built the model for mFloram, we sought to get as much information as possible from its genome sequence and to challenge the current annotations since, as we mentioned, one of the main goals in minimal cell research is to reach complete knowledge of molecular functions. To do so, we use a combination of three computational approaches, namely proteome comparison, structural homology, and EC number identification. Here, the example is given for the enolase of mFloram. So first, what we do is to compare the proteome of mFloram with that of 4 other molecules, for which metabolic models were previously generated. Then we use the ITaster software to reconstruct reliable 3D structures that could be used to predict functions. And finally, we use the DTEC software to make probabilistic identification of EC numbers, which are very useful for metabolic reconstructions. Combining this information allowed to trace the reliability of gene annotation across the genome. Here, the top layer in blue shows a higher level of confidence in each method, whereas the bottom in red shows a lower level of confidence. In most of the cases where no gene names were identified, we also see that no protein structures or EC numbers could be determined. Using this combination of methods, we are also able to target which annotated genes may be less reliable than one could originally think. This provides some valuable information for future biochemical studies. So here is the product of the reconstruction, the entire map of the metabolic network of Mesoplasma florum. In itself, I think it is very interesting to consider that the entire metabolic map of an organism can fit on a single slide. This is definitely not something you could say for a human cell or even E. coli. The complete network is composed of 208 genes and roughly 350 reactions. Given the small size of that network, you may wonder how an organism manages to live with so few elements. Here are a list of features we learned from this reconstruction. First, Florum uses only glycolysis for energy production. This mode is not the most cost efficient, but avoids a heavy gene burden that is associated with the cellular respiration. In fact, there is no need for an electron transport chain or having a second cell wall to create the electron gradient. Second, studying the pathways in mFloram reveals that anabolism is nearly absent. So one way to cope for that lack is to recycle existing components. A good example of this feature is the nucleotide salvage pathway, which is one of its most complex pathways, but allows the interconversion of many nucleotides with one another. So a lot of recycling happening. Finally, most of its building blocks are taken from its growth environment. It is noteworthy to consider that the genome complexity gets transferred from the number of genes to the medium complexity. So key metabolites like amino acids and lipids are imported directly from the medium, and no synthesis is seen in the mFloram network. We then used above that software to define a species-specific biomass objective function for Mesoplasma florum. As you can see, we used the macromolecular wave fractions defined previously in our integrative characterization study with multiple omic data sets to define which metabolites should be added in the biomass objective function and in which proportion. As we mentioned, the complexity of mFloram is transferred to its growth medium. The original medium was the ATCC 1161 and is composed of undefined components such as yeast extract and serum. As you can see on the left, this medium actually allowed growth when no sucrose was added. This was problematic because we could not alter the medium to test any growth phenotypes. We found that supplementing with CMRL 1066 allowed to simplify the medium by reducing the concentration of undefined components, reaching a level where no growth was detected when sucrose was added. This CSY medium, as we call it, enabled the definition of key uptake and secretion rates that could be used as constraints on the model, as you can see on the right. These panels show the sensitivity of the model to different constraints. Here, the sucrose uptake as well as the lactate and acetate secretion rates were obtained from experimental growth data on CSY medium. Interestingly, the experimental sucrose uptake reaches a plateau at a rather low rate, which is consistent with the low energy requirement of mFlora. The CSY medium also allowed to validate the assimilation of different carbohydrates by mFlora. Here we tested the assimilation of 14 different carbohydrates and found that both maltose and mannose allowed growth while it was not predicted by the model. This provided a context to reevaluate the alternate carbon metabolism of mFlora and suggest promiscuous reactions. To address this, we compared the 3D structures generated by ITASR to structures in the protein databank for which promiscuity is known. Using the FATCAT software, we found that the 3-allose-6-phosphate hydrolase, MFL499, was actually very similar to an enzyme known to also hydrolase maltose 6-phosphate. Comparing the phosphoglucose isomerase, MFL254, to an enzyme capable of converting mannose 6-phosphate into fructose 6-phosphate also revealed a similarity. Together, the experimental validation of sugar assimilation with the structural comparison provides testable hypotheses for missing biochemical functions in mFlora. As seen on the blue heat map, we compared the model flux states predictions with gene expression obtained from proteomic and transcriptomic datasets, and we were able to get an accuracy of 78%. Similar comparison with single gene essentiality predictions against transposon insertion data revealed a 76% accuracy, as can be seen on the green and red heat map. These comparisons revealed false negative predictions that could be manually examined. An interesting case is that of the ribulose phosphate epimerase. Since the transaldolase of the pentose phosphate pathway is generally missing in molecules, the model could not use this gene. To resolve this false negative, we sought to find an enzyme that could act as a transaldolase in mFlora. Using FATCAT, we found a good structural match between the aldolase MFL121 and a known transaldolase in the protein databank. We therefore provide a candidate for a future biochemical study that could elucidate the use of pentose phosphate pathways in molecules. We then use the min genome algorithm to formulate a minimal genome prediction for mFlora. The algorithm takes as input the model, genome-wide essentiality data from TNSeq, and the transcription architecture. You can see that by performing iterative deletions, both the genome size and the number of genes reach an asymptote at 75 deletions. The final genome size is 552 kilobase pair, which is very similar to JCV-ISIN3 at 531 kilobase pair. The number of deleted protein coding genes goes up to 161, bringing the total of proteins in the prediction to 519, which compares to the gene count of 473 in JCV-ISIN3. Finally, 57 mFloran genes were predicted to be deleted while having orthologs in JCV-ISIN3. This suggests a potential difference in the minimal genomes. To understand the functions entailed within that minimal genome prediction, we represented the expressed protein on the Voronoi diagram on the left. On this diagram, dark areas represent the proteins that were predicted to be deleted by the genome. The first feature we realize here is that most of these proteins are in the unknown category, which fits our objective to reach complete understanding of molecular functions, meaning that by reducing the genome, we would mainly remove proteins of unknown function. The metabolic category comes second in total gene count. Most of the metabolic proteins that were removed here are in the PDS system or transport. This means that, metabolically speaking, the first targets of genome reduction could be in the alternate carbon metabolism. Finally, the genetic information processing comes third. This category was not modeled, so the prediction of gene deletions are relying only on the single gene essentiality obtained from TNSeq data. Obviously, this could be an issue because of the possibility of synthetic lethality. Which brings us to our perspectives. At the beginning of the presentation, I asked three questions. First, what do we learn as we model the metabolism of mFlora? We've seen that its metabolism is based on salvage and depends heavily on import from the growth medium. It also has a small energy requirements. Second, we wonder if we could make a minimal genome prediction using this model, which we did, and yielded a minimal gene set that contains 519 proteins and a genome size of 552 kilobase pair. Since mFlora is phylogenetically close to the synthetic minimal cell JCV-ISIN3, We can compare the prediction of this minimal gene set to this bacterium. We saw that 57 genes were targeted by deletions that were also orthologs to JCBISN3. This suggests that the minimal genome is not a unique solution. Finally, I would like to conclude by saying that our current efforts have leveraged experimental gene essentiality with metabolic model to formulate the prediction. This prediction is interesting, but is obviously limited by the capacity to address synthetic lethality outside of the scope of the model. Therefore, using a model of both metabolism and gene expression would probably yield a more reliable prediction. I would like to thank the two research groups that supported this project, so the laboratory of Professor Sébastien Rodrigue at the University of Sherbrooke, and the Systems Biology Research Group led by Professor Bernard Paulson at the University of California in San Diego, that I had the chance to visit for a few years during my PhD. Also, thank you to the founding agency that supported the project, and I will be ready to take your questions now.